Good afternoon, everyone. I am your yeah. Good afternoon, everyone. I am your host Alok Kumar Jha. We will begin the webinar in. Welcome you to today's webinar on a fascinating topic: exploring EGFR TK inhibitors binding to various EGFR mutations using non-covalent and covalent docking in ICM. We are thrilled to have you join us and look forward to an engaging session. Before I dive into the topic, let me take a moment and introduce you to Advent Informatics. Advent Informatics Private Limited is a Pune-based distributor of life science softwares. We provide software solutions in the domain of chemoinformatics, molecular modeling, medicinal chemistry, chemical data curation, and chemical and biological data management to most of the major pharma, chemical, and CRO companies in India. We also work with major academic and government research organizations and focus on providing well-renowned software to Indian researchers and students. We currently represent Molsoft LLC, Chemexon KFT, Chempas, Certara, Transgen IT Solutions, and Key Module. Now let's dive into today's session. EGFR mutation plays a pivotal role in cancer research, and understanding inhibitor binding through docking studies is a critical area of focus. Today, we will explore the binding interaction of non-covalent and covalent EGFR TK inhibitors using docking approaches in ICM for analyzing EGFR mutations, a topic that holds immense relevance in drug designing and molecular modeling. Now, let me introduce you to our speaker, Dr. Pathan Mohsin Khan. Dr. Khan has been an application scientist at Advent Informatics Private Limited for about 1.5 years now. Prior to joining the company, he worked as a senior R&D specialist at QSAR Labs and interned as a research data scientist at Edelweiss Connect, Basel, Switzerland. Dr. Khan holds both a doctorate and a postgraduate degree from the National Institute of Pharmaceutical Education and Research, that is NIPER, Kolkata. Demonstrating a solid academic background, he has published 12 research articles in peer-reviewed journals and 3 book chapters with renowned publishing houses. He is proficient in structure-based and ligand-based drug design techniques, encompassing protein modeling, binding site predictions, molecular docking, pharmacophore modeling, QSAR modeling, virtual screening, and many more. If anyone has questions they want to ask to Dr. Khan during his talk, please submit them in the Q&A section. You can find the Q&A option in the toolbar at the bottom of your screen. Dr. Khan, the floor is yours.
Thank you, Alok. Hope all of you are able to hear me. Good afternoon, everyone. Thank you for joining us for today's webinar cum demonstration on the topic of exploring EGFR tyrosine kinase inhibitors binding to various EGFR mutation using non-covalent as well as covalent docking in ICM. So what is EGFR tyrosine kinase inhibitors and why they are important? EGFR tyrosine kinase inhibitor play an important role in targeted cancer therapy, especially in the treatment of EGFR mutated cancer. And this is a rapidly evolving field, and understanding how different EGFR mutation interact with different non-covalent as well as covalent inhibitor can significantly add in rational drug design against this particular EGFR protein. Before we will move deep dive into EGFR inhibitors, let's talk about basic history as well as the basics of covalent inhibitors. So let's move into the history of covalent inhibitors. So the history of covalent inhibitors is quite fascinating, and it all started in early 90s, where some of the first covalent drug which were discovered by serendipity, like aspirin and penicillin, which was discovered by chance, and they act through the covalent they act through the formation of covalent bond with their target receptor protein. And it has increased the interest of researcher to develop novel covalent inhibitor in the mid 20th century. However, due to different side effect as well as toxicity and lack of selectivity of these covalent inhibitors towards the target, the interest to develop novel covalent inhibitors has been declined, and this era will be known as the decline phase for covalent inhibitors or covalent drug molecules. In the next era, in the next phase, you can say there was advancement in structural biology techniques like X-ray crystallography and protein modeling, which will provide atomic level information regarding the protein ligand interaction. At the same time, there was advancement in the computational tools which will provide better understanding of covalent bond formation between protein and the ligand molecule. And there was also advancement in the field of in the field of understanding the selectivity of warhead and the residues, reactivity of the residues which are involved in the covalent bond formation, which has increased the selectivity and reduces the toxicity or side effect of those covalent inhibitors. Then in the latest decade, which will be known as a golden era for covalent drug, in which there are several covalent inhibitors has been approved by FDA, like ibrutinib, afatinib, which has been approved and demonstrating the potential of covalent inhibitors in the market. As you can see in the image, from like 1899 to 2021, there are several well-known US FDA approved covalent inhibitors which are present in the market, like from aspirin, penicillin. Then here you can see ibrutinib, osmartinib, afatinib, which we will discuss in this demonstration webinar come demonstration session. Now let's move toward the what is covalent docking or what is covalent inhibitors. So covalent inhibitors basically which will form a covalent interaction with the targeted protein, and which is a irreversible complex which will provide a prolonged effect of that particular molecule toward the particular target. Now let's move toward the covalent docking. What is covalent docking? So covalent docking is a computational method by which one can predict the binding of small molecule to a macromolecule, which will be either protein or RNA, through the formation of covalent bond. And this covalent bond formation is very important in drug design, as this interaction can lead to reversible as well as irreversible inhibition of target enzyme. Now, if you will see at if you will look at the process of covalent bond formation or covalent docking, which is divided into two steps. One is non-covalent association. In the first step, the ligand molecule initially interact with the protein through non-covalent interactions like hydrogen bond interaction, electrostatic interaction, van der Waal, hydrophobic interaction, and it will place the ligand molecule inside the binding pocket of the protein of interest. And the warhead, which is a reactive functional group in the ligand molecule, is will be kept in close proximity to the reactive residues present in the ligand molecule. And in the second step, the covalent bond formation between the reactive group in the ligand molecule and the nucleophilic residues present in the binding site of protein of interest, they will form a covalent bond and form a complex. So if you will see the energy diagram for this particular covalent bond, covalent docking, in the first step, if you will see, the ligand molecule and the protein are in the free state, which have a lower energy. But when they will form a non-covalent complex, they will require some sort of energy, and after that particular some sort of energy, and they will form a non-covalent complex which will be stable at this particular position. Then after formation of this non-covalent complex, and they will to form a covalent bond between the reactive warhead and the reactive residues present in the binding site, it will again require the energy, which will increase the energy diagram. And then after formation of covalent bond, it will again reduces the binding energy for binding energy of the stable complex, which have covalent bond formation between the protein and the ligand molecule. Now let's understand why covalent docking is important. So the main advantage of covalent docking lies in its ability, like it will enhance the potency by forming a stronger or long-lasting effect due to irreversible or stable binding to the target. It will also improve the selectivity by designing proper warhead and scaffold, ensure high specificity and reducing off targets. It will also facilitate to understand biological and biochemical pathway and protein which are function through stable protein ligand complexes. And similarly, covalent docking or covalent inhibitors are also very important for targeting traditionally undruggable target, like recently KRAS, which is a well-known protein, which was previously considered as a undruggable target. But now, recently, sotorasib, which is a covalent inhibitor for that particular target, has been designed by using covalent technologies. Now, let's move toward the key concept or key parameters which are required for designing of covalent inhibitors. The first one is a key component which required for designing a covalent inhibitor is a reactive group in the ligand molecule, which is also known as a warhead. So this warhead is a reactive functional group on a small molecule that enable it to form a covalent bond with a specific nucleophilic residue in the target protein. And there are several warhead so far has been explored and also present in the several FDA approved drug. Like you can see beta lactam warhead, which is present in different beta lactamase beta lactam antibiotics like penicillin. Then similarly, you can see acrylamide, which is present in ibrutinib as well as afatinib. Then there are other warhead like cyanoacrylamide, then epoxide, aziridine, then boronic acid. And so there are a number of warhead as of now has been explored by a research community. There are several parameters which one has to keep in mind prior to a design, prior to select the warhead. One is balance of reactivity. The warhead must be reactive enough to form a covalent bond with the reactive residue, and it should not be so much reactive that it form off target or premature bond with other target and causes side effects. And that particular warhead should be electrophilic. Most warhead act as a electrophile, and they will target to the nucleophilic residues. Therefore it should be electrophilic. Then selectivity and the structural context, you can see the geometry of the particular warhead should be complementary with the binding site of protein of interest. And then stability, and the warhead must be stable throughout the biological transformation. Now let's move toward the another concept which is more important in covalent inhibitor designing, which is a reactive residue. So reactive residue basically refer to a specific amino acid within the target protein that contain a nucleophilic functional group capable of forming covalent bond with a electrophilic warhead on the ligand molecule. And so far there are five different residues which has been targeted by research community, like cysteine, serine, threonine, histidine, and lysine. And all these residues have a nucleophilic functional group. Like in cysteine you can have a thiolate functional group. In serine and threonine you have a hydroxyl functional group. Then in lysine amino group, and in histidine imidazole is acting as a nucleophilic functional group which can react with the reactive warhead of the ligand molecule. Now to there are several factors which also affect the reactivity of each of the residues, like proximity to the ligand molecule. The residues which are present in the binding site should have a close proximity toward the ligand molecule, so that it can form a covalent bond interaction with the ligand molecule. Also the orientation and accessibility of that particular reactive residue, it should be on the surface or within the binding site, so that it can interact with the ligand electrophilic groups. And the environmental factor also affect the reactivity of residues, like pH and solvent condition can significantly affect the reactivity of amino acids present in the binding site. Let's say for at higher pH, cysteine is highly reactive, but at neutral pH, serine and threonine will have a higher reactivity. Similarly, steric hindrance, and the steric hindrance should not be there in the binding pocket, so that it will not allow reactive residue to form a covalent bond with the reactive warhead of the ligand molecule. And then some electrostatic environmental factors. Now let's move toward the different ways by which we can perform covalent docking in different softwares. So there are different ways by which one can simulate covalent bond formation or covalent docking in computer. The first one is a direct linking approach. So in this approach, one can form covalent bond prior to covalent docking between the reactive warhead and the reactive residue and the warhead of the ligand molecule. And this particular approach is used in ICM for covalent docking. Then another approach is a variant of direct linking approach, in which, like, either there are several approaches, like one can mutate the reactive residue in the binding site of protein of interest to some other residues, and then place the ligand molecule in, inside the binding pocket of the protein of interest, and then again remodify the modified residue with the original reactive residue, and then form a covalent bond and perform refinement of that particular complex. And the third approach is hybrid approach, which is based on quantum mechanics as well as molecular mechanic calculation. So by which one can more accurately model the covalent bond formation between the protein and the ligand molecule. Now, which approach one should use for performing covalent docking? So for that, one has to answer for several questions, like level of available, highly content. The challenge in selection of covalent docking approach is highly context dependent and varying based on level of available knowledge and the specific objective for the targeted system. So depending on the answer of some particular questions, one can address which approach one can use for covalent docking. The first question is the binding site of target protein is known? If the binding site of the target protein is known, then then you have the answer for that particular protein of your interest. Then is the target amino acid identified and its reactivity is understood? If that is also understood, then you can use the simple approach. Similarly, is the electrophilic warhead of the ligand molecule is also defined, which warhead is reacting with the residue. You can also if you know the warhead also, then you can use a simple approach. Then last question is what level of accuracy is needed for affinity as well as reactivity prediction. So there are several questions here. You can see, is the binding site of protein of the target protein is known? So if you don't have the known information about the binding site, in that case you can use ICM pocket finder to identify binding site. Similarly, is the target amino acid identified and its reactivity understood? If the amino acid, there are five different amino acids so far has been explored, you can from visual analysis you can see which residues are present in your binding site. And there is a approach or feature in Molsoft which can predict the reactivity of only cysteine residues by using a random forest model as of now. Then you can have, if you have already known information about the electrophilic warhead, then you can use this similar molecule which contain the similar warhead, different molecule with similar warhead to dock within the binding site. So for that you just need a simple direct linking approach. But in a case where you have to identify the reactivity of scaffold, reactivity of the warhead, as well as you need a high level of accuracy, then in those cases you require a hybrid approach, like quantum mechanics and molecular mechanics approach, which is a time consuming and long process. So now let's move toward the how one can predict the reactivity of cysteine residues in their protein of interest in Molsoft. So we have a features which can predict the reactivity of cysteine residues in the protein of interest. So this a particular feature is based on random forest model, which is built on the data set of reactive as well as non-reactive cysteine, and data set of reactive as well as non-reactive cysteine residues present in the molecule, present in the protein structure. And the random forest model will predict the reactivity score for each of the cysteine residues present in your protein of interest, by considering several parameters or several features like solvent accessible surface area of cysteine, side chain, pocket area, if there is a binding pocket within the 2 angstrom of this cysteine, then distance to nearest positively charged lysines or arginine residues, and all of other parameters which are which are mentioned over here. And this particular model is trained on the data set on the training data set, and validated on the test data set, and the accuracy for a test data set is on more than 95 more than 93%, 90%. And similarly, if the reactivity score for each of the residue is greater than and equal to 0.06, will be considered as a reactive cysteine present in your protein of interest. So I will just quickly show how we can use this particular feature in feature to predict the reactivity of cysteine residue in any protein by using Molsoft. So I'll just open up the Molsoft. Here I have Molsoft. So, and I will import a protein structure which is complex with the covalent ligand molecule. So the PDB ID for that particular protein structure is 4G5J. So here we can see we have a protein structure which is complex with the ligand molecule. Here you can see it is a afatinib, which is complex with the EGFR protein. I will show you the interaction also. You can see over here this cysteine 79197 is forming covalent bond interaction with the ligand molecule, which is afatinib. Now we will remove this ligand molecule from the same object. Now we have only the single objects which have protein structure. Now we will predict the reactivity of each of the each of the cysteine residues in this particular protein structure. So for that, you just have to go to this tools option here. In the tool, you have this 3D predict, and in the 3D predict, you have this reactive cysteine option. And here you have to select the particular object of your protein structure to predict the reactivity of cysteine. It will you can see, and the title of this particular window is identify reactive cysteine residues in this particular set of residues. You just have to press OK, and it will start predicting or identifying different residues based on that random forest model. And after completion of the job, you will have a list of all the residues present in your protein structure, whether it is a reactive or whether it is a non-reactive, in the tabular form. So let's wait until it will finish. Till then, we will go ahead with our presentation. Now let's move toward the approach which we will use for covalent docking in Molsoft. So the approach which we will use for covalent docking in Molsoft is similar to non-covalent docking, and just one extra step, which is you have to specify your reactive residue and specify the reaction model. So the reaction model basically describe the transformation of a receptor side chain which will form covalent bond interaction with the warhead of the ligand molecule. And rest of the step you will see are automated, but I have mentioned it over here. Like side chain atoms which are involved in the reaction are masked in the docking grid, as well as ligand, or whatever ligand molecule you will dock by using this approach will automatically transform or align with the reaction model which we have used Then docking constraint, some side chain atom and atom coordinates are used to constrain the placement of ligand molecule inside the binding pocket of interest, and the conformation of the ligand molecule is generated by using Monte Carlo simulation method. And finally, all the generated conformation were scored based on the different ICM scoring function like ICM score as well as RTCN score and MFE score. Now let's move toward the database of common covalent reaction transformation models. So in Molsoft, we have almost approximately 80 plus prepared reaction which cover different residues like cysteine, serine, histidine, threonine. So all these reaction transformation are already available in the Molsoft. If your warhead which is forming covalent bond interaction with the particular residue of interest in your protein structure is not available in the database, you can draw your own chemical reaction with the warhead and the reactive residue, and you can use that particular chemical transformation or reaction transformation for docking of your own covalent ligand molecules. Now let's move toward the benchmark study of covalent docking in Molsoft. So the benchmark study was performed by Professor Catrich and Eugene and uh, Professor Catrich and Maxim Totro to identify small molecule inhibitor targeting ubiquitin like pox virus, proteinase 17L through homology modeling as well as covalent docking. So first of all, they have built a homology model by using Molsoft for that particular protein of interest, and then identified the binding site, and then they have used the covalent docking approach to virtually screen 3.5 million compounds. And after virtual screening of 3.5 million compounds, they have identified 450 candidate inhibitors, and those 450 candidate inhibitors were substituted for biochemical assay, and then found out that 97 compounds out of 450 are showing inhibitory activity against that particular pox virus proteinase 7L enzyme. So for this particular covalent particular benchmark study, they have used four different types of warhead like aldehyde, aliphatic aldehyde, aliphatic aromatic aldehyde, aliphatic ketone, as well as aromatic ketone. You can see the library of all those libraries of all the warheads, and how many compounds from each of the warhead has been selected based on the ICM docking score. And out of those identified hits, how many of them has been tested positive and shows activity in the biological assay has been reported in this particular table. Now let's move toward the another success story by using covalent docking, which is FDA approved drug Webomir. Webomir is basically a combination of two drugs. One is meropenem and another one is verbobectam. So verbobectam is a cyclic boronylated compound which was initially designed and developed at Molsoft. And this cyclic boronylated compound is forming a covalent bond interaction with class A serine carbapenemase enzyme, and due to that particular formation of covalent bond and high affinity toward the high selectivity toward the particular target of a particular class of enzyme, it was prioritized for synthesis as well as biological evolution, and in collaboration with the Rampex pharmaceutical in combination with meropenem, it was granted FDA approval in 2017. And this success story is already published in Journal of Medicinal Chemistry in 2015. Now let's move toward the comparative study of covalent docking tool. So one of the independent evolution which was published in Journal of Chemical Information and Modeling in 2018, in which they have compared six different covalent docking tool from freely available as well as commercially available software tools, and they have found out that ICM Pro is performing very well in comparison with well-known freely available as well as commercially available covalent docking tool in terms of reproducing the experimental binding mode as well as scoring the generated binding poses. They have also highlighted that if we will have a more challenging ligand molecule or more flexible ligand molecules containing more than 35 heavy atoms, then ICM perform better than any other freely as well as commercially available software tools. They have also highlighted that if you will use non-covalent conventional docking of Molsoft, which will also give remarkable rate of accurate prediction for covalent inhibitors. Now let's move toward the today's demonstration case study, which is EGFR or epidermal growth factor receptor. So this particular receptor is a tyrosine kinase receptor, which is encoded by EGFR gene on chromosome. And this EGFR is activated by binding to a ligand, triggering downstream signaling pathway involved in cell growth, proliferation, differentiation, and survival. So this particular activation of this particular EGFR leads to cell proliferation, which is involved in the cancer. So now we will see which are the inhibitors which are targeting this particular EGFR protein. So there are several generation of three generation of inhibitors which are designed to target the EGFR protein. The first generation are covalent non-covalent inhibitors, which are simple inhibitors like gefitinib, then erlotinib. And this particular first generation of inhibitors have some limitation, like they have acquired resistance, like some mutated EGFR protein, which is T790M, which is threonine 790 methionine mutation, which will increase the affinity towards the ATP binding and reduce the affinity or binding for first generation inhibitors. And also this first generation of inhibitors have some side effect like skin rashes and diarrhea. And due to non-covalent inhibitors, they are also forming reversible complex with the protein of interest, which result in less sustained inhibition of EGFR activity. So there was a need to develop the second generation of inhibitors, and the scientists or researcher have focused on designing second generation of covalent inhibitors with second generation of inhibitors which can form a covalent bond interaction with the protein of interest. And they have targeted the cysteine they have targeted cysteine 797 residue in the protein, in the binding site of protein, to design a second generation of covalent inhibitor, which is afatinib and docomitinib. However, this second generation of inhibitors have several advantages over the first generation, like it will form a irreversible binding with the ligand with the receptor protein, which will provide longer lasting inhibition of EGFR activity. Also, this particular second generation of inhibitors are also uh, uh, are more effective against some mutations, but they are less effective or limited efficacy against the threonine 790 mutation, methionine mutation, while slightly better than they are slightly better than the first generation of tyrosine kinase inhibitor. However, they are still ineffective against this particular type of mutation of this particular EGFR receptor. And they have also limited limitation like diarrhea and skin rashes. So there was a need to develop a third generation of inhibitors which have a higher selectivity towards this particular mutation, which is threonine 790, which is mutated to this threonine mutated to methionine. So there was a need to develop a third generation of inhibitors, and then researcher has come up with a new molecule, which is osimertinib, which is effective against this particular mutation of the EGFR protein. Similarly, it has improved selectivity over the, like, it will more selective toward the mutant EGFR than the wild type of VGFR, which will reduce its side effect. But it's still this third generation of EGFR inhibitors are also having its own limitation, like it will induce the 797 cysteine mutation can be developed by using if one can use this third generation of inhibitors, and they are highly expensive. So I believe this is it from the presentation. Now we can see how this first generation, second generation, and third generation of inhibitors are forming interaction with the EGFR proteins. And we will explore how we can perform covalent docking by using second or third generation of EGFR protein in Molsoft. If you have any questions prior to that, you can ask me. So far I will, as you can see, it has determined the reactivity of each of the residues present in this particular protein structure. And the reactivity of each of the residues are reported in this particular table. And you can see the score, which is a reactivity score. You can sort this table. You can see over here the cysteine 797, which is having highest reactivity, which will be considered as a reactive residue in this particular protein structure. And at the same time, if we have seen that this afatinib is also forming covalent bond interaction with the cysteine residue or cysteine 797 residues, which is present over here. You can see 797 residue, which is forming covalent bond with the acrylamide warhead of this particular afatinib molecule. Is there any question so far? So I'll just clear the session. So now we will import a first generation of EGFR inhibitor, which is complex with the EGFR protein. So the PDB-ID for that particular protein structure is 4WKQ. And I'll just import this particular protein structure. As you can see, this is a first generation non-covalent inhibitor, which is gefitinib. As you can see over here in the workspace panel, I will remove the unnecessary units. If you will see, and this is a first generation of non-covalent first generation EGFR inhibitor. You can visualize the interaction of this particular ligand molecule. Just right click on the ligand, go to pocket and ligand pocket interaction, and just zoom in. You can see this particular ligand molecule, which is gefitinib, is forming non-covalent interaction with the protein of interest. And this particular complex structure, if you will see here, this threonine 790, if you will see this threonine 790, if you will see this threonine 790, this is a residue which is not mutated as of now. So it is forming covalent bond non-covalent complex and accommodating within the binding site of protein of interest. However, if you will mutate this particular residue with the methionine, so you can see this particular chlorofluoro phenyl ring is forming clashes, will form steric clashes with the side chain of methionine residue. So let's explore how it can form steric clashes with the methionine residue. So now we are mutating this threonine 790 residue with the methionine. To mutate this residue, you just have to right click on the particular select the particular residue, right click and go to advanced, and in advanced you will see the option. If you will see here, advanced mutate amino acid, and we will mutate this particular amino acid with the methionine, which is a resistance for this particular class of inhibitors, methionine, and just press OK. And now you can see we have modified the threonine with the methionine. And if you will see, the clashes between this particular chlorofluorophenyl and the side chain of the methionine residue, for that you just have to go to this label tab over here, and you will see this option here, toggle clashes, and it will show you the clashes between your ligand molecule and the protein of interest. So you can see this particular part of the ligand molecule is forming a clashes with the binding site residue of the protein of interest. However, these particular clashes are not available in the original or wild type of protein of interest. These particular clashes is available only in the mutated protein, which is a mutated protein, mutated EGFR. So this particular side chain, which is bulky and more hydrophobic than the threonine, form steric clashes with the first generation as well as second generation of inhibitors, and they are not able to form a stable complex uh, with the with the mutated protein, and have a less affinity than the ATP molecule. Similarly, if you will see the second generation, I will just import the second generation structure also. Let's just search second generation, which is 4G5J import. So this is our second generation protein. You can see here, which is afatinib. I will just show the covalent bond interaction also. So now you can see this is afatinib, which is forming covalent bond interaction with the cysteine 797 residue. And in this particular protein structure also, you can see this is threonine 790, which is wild type of protein. But if you will mutate this particular particular residue with the threonine or methionine, let's just mutate in this particular structure also. In this particular conformation of the ligand molecule, if you will see the clashes, there won't be any clashes between the protein and the ligand molecule. You can see, and there won't be any clashes between the protein and the ligand molecule. So they are effective against the wild type of EGFR. However, if we will mutate this particular residue, this threonine 792 methionine, right click, advance, and you go to mutate amino acid and change it to methionine. And then if you will click on this toggle clashes, sorry, yeah, toggle clashes, you will see you will see all the clashes between the threonine side chain and the particular chlorofluorophenyl ring of the afatinib molecule is forming a steric clashes with the receptor protein, and it will reduces the affinity of this particular second generation covalent inhibitor in the binding site of a mutated protein. But now let's see, we will import the third generation of inhibitor, which is the PDB-ID for the third generation of covalent inhibitor is 7JXP. So in this particular, I will remove the unnecessary chain. In the C chain, we have a covalent inhibitor, which is so here you can see this is our third generation inhibitor, which is osimertinib. So if you will see the interaction of this particular osimertinib with the EGFR protein, right click, and so now if you will see in this particular protein structure, the already methionine is there in the binding site of protein of interest. So this is a mutant EGFR in which say threonine is mutated to methionine, and you can see this particular particular ligand molecule is forming covalent bond as well as some non-covalent interaction with the target protein, and it is having higher affinity towards the mutant EGFR protein than the first and second generation inhibitors, because it is not having any clash, it is not forming any steric clashes with the mutated residues in the binding site, and it will also shape complementary with the binding site of protein of interest, and it has highest affinity than the ATP molecule towards this mutated EGFR protein. Now if you will superimpose all this particular structure, just select all the structures and click on this button here, and you can see superimpose on particular one protein structure. Now if you will see, just zoom out, you can see over here in each of the third generation inhibitor, which is in blue color, you can see over here, we can remove the first generation and second generation. Yes. So you can see over here this in both the protein, this is the methionine 790, and in our mutated protein it is 719, and in both other protein it is threonine 790 residue which is present in the binding site. And if you will see, if you will compare the binding pose of each of the first generation with the third generation, so you can see the third generation is third generation indole ring is present outside the binding pocket. However, in the first generation, this chlorofluorophenyl ring is forming steric clashes with the methionine 719 790 residue. Similarly, if you will compare with the second generation also, it will also forming clashes with the methionine residue, methionine 792 790 residue in the binding site of protein of interest. Now let's see how we can perform small molecule non-covalent docking and covalent docking in Molsoft. So for non-covalent docking, it's a pretty simple approach. I will just show you how we can perform covalent docking for covalent docking for this third generation of inhibitor. So I will just convert this particular protein structure into ICM object. So whatever object you have to convert that particular object into ICM object. As you can see, the job has been started. And after completion of the job, it will update our X-ray structure with the prepared one, which is ICM object. And now in this particular object, we have a osimertinib, which is in complex with the ligand molecule. If you will see here, ligand interaction. Just so now you can see over here this cysteine 797 residues forming covalent bond interaction with our osimertinib molecule, which is a third generation molecule. So now we will remove or move this ligand molecule from the same object. So to remove this ligand molecule, you just have to right click and move from the object, and it will create a separate object for the ligand molecule. Now we have a mutated protein, EGFR protein in 7JXP object, and we have a ligand osimertinib ligand molecule in this particular object. We can directly extract the 2D structure of this ligand molecule. Right click and extract 2D structure in a chemical table. Now we have a 2D structure of this ligand molecule in a chemical table. You can see this is the warhead, this acrylamide warhead present in this third generation of EGFR protein. So now we have to generate a grid map considering only this particular protein structure. So to generate a grid map, you just have to go to docking, select new project, and in the new project you can just provide name like Covalent, and then select the particular object in which you want to generate a grid map. So we want to use this particular object or this particular protein structure as our receptor 7JXP. So we'll just have to select the 7JXP, and we will define the binding site around the co-crystallized ligand molecule which is present in this particular complex. So we'll select that particular ligand molecule and click on this define site around selected molecule. And in simple to generate a grid map for simple docking, we will keep this option here, make receptor map immediately, always check. But in a covalent docking or induced fit docking, you have to uncheck this option here, because in covalent docking and in induced fit docking, we have to again remake the generated map by considering the reactive residue as well as the reaction transformation. Or in case of 4D docking, we have to consider multiple conformation to generate a grid map in the later on stages. So you just have to uncheck this option here and then press OK. It will generate a sphere at the binding site of at the center of the binding site. You just have to press OK again, and it will generate a purple grid box. You just have to again press OK, and it will start generating a grid map. And after generating a grid map, it will add that particular generated grid map within the workspace panel with the name which we have given to our setup receptor project. Now we have this particular object. So in this particular object or generated grid map, we have to select the particular residues which are involved in the formation of covalent bond. So in our case, in this particular protein structure, cysteine 797, which is over here, you can see is involved in formation of covalent bond. So you just have to expand the sequence of your protein structure, sequence of your protein in the generated grid map, and select the particular residue which is involved in the covalent bond formation, like cysteine 797. Then go to docking, and in the docking you will see this option here, covalent docking. You just have to you just have to click on this option here, covalent docking, and it will open up this window. So you just have to uncheck and select the first residue which is involved. Here you can see the name of your grid file, which is my.doc covalent, and in this grid file you have to select the reactive residue. So here you can see 797, and this is 797. Graphical selection, one residue has been selected. Now you have to select the reaction transformation by which your covalent ligand molecule will form a covalent bond interaction with the reactive residue of the protein of interest. So by default, we have already more than 100, more than 80 reaction transformation available in Molsoft. So you just have to click on this load preset reaction, and it will provide you list of all the available chemical transformation by which reactive residue or reactive warhead form a covalent bond. So in our case, the reactive warhead or warhead in the ligand molecule is acrylamide, and the reactive residue is cysteine. So we just have to search that particular combination in this particular reaction database, and you can see here acrylamide and cysteine. So this acrylamide warhead is forming covalent bond with interaction with the cysteine residue. So you just have to select that particular chemical transformation and add to docking project, and it will automatically add that particular reaction transformation into to your grid map. And then you will see this option here, remake receptor map immediately, and you just have to press OK, and it will start generating a grid map again for your protein of interest by considering this reactive residue as well as the reaction transformation of your covalent bond formation. And after this final generation of final covalent bond, final generation of your grid map, then you can directly dock your covalent ligand molecule. This is our covalent ligand molecule. Now we want to dock this covalent ligand molecule. You just have to go to docking. If you have more than 100 compounds, then you can use this setup batch ligand docking. If you have a single ligand or more less than 100 compounds, then you can use this dock chemical table option here and select the particular table which you want to dock. So we want to dock this, redock this particular ligand molecule, and just you can check some you can edit some default parameters. For that, you just have to uncheck this option here, dock immediately, and press OK, and it will open up this window, tools window over here. So here you can generate 10 conformation if you want to generate 10 poses for your ligand molecule. Similarly, if you want to sample double bond present in your ligand molecule into cis and trans configuration, sample racemic center into cis and racemic center into RNS configuration, sample tautomers, and you just have to press dock, and it will start your covalent docking for this particular ligand molecule. And after completion of the job, you will have a notification window in which you have a status of the job, whether it is completed normally or whether it is completed with some sort of error. And it will ask you whether you want to import the generated results or whether you want to save the generated results for later on visual analysis as well as statistical analysis. So as of now, if you will see this particular protein structure or generated grid map is represented in wire representation. You can change it to ribbon representation from here. You can hide wire representation. You can remove the transparency of your protein structure from here. So let's wait until it will finish. If you have any questions so far, you can ask me.
Yes, Dr. Khan, we in fact have many questions, but we will address them after the session is over.
Okay. So I'll just show you the results also. I have already performed for third generation, second generation. So I'll just webinar covalent docking of third generation. So as you can see, we have a co-crystallized ligand molecule here, which is 7JXP_1, which is in green color. And we have a co-crystal generated poses for this third generation inhibitor in the chem_doc table. You can see over here. And each of the warhead, acrylamide warhead, is forming covalent bond interaction with the cysteine 797 residue. You can see in the result table also in the mole column. Now, if you want to import the first pose of the covalent inhibitor, so you just have to double click and it will import that particular covalent particular ligand molecule inside the binding pocket of your protein of interest. And at the same time, it will generate that particular complex within the same grid map, like mydoc_receptor. Previously we had only the protein structure, now we have a ligand molecule as well. But now if you will see in this particular visualization, we are not able to see which residues are forming different types of interaction with the ligand molecule. So if you want to generate a better visualization, so you just have to click on this setting button over here and go to tools. In the tools, if you will go slightly lower, you will have this visualization option here. In the visualization, there is a visualize button, and in visualize, and there are multiple options in which you can visualize your protein ligand complex. One of them is pretty. You just have to select that and click on visualize, and it will generate a pretty view visualization for your protein ligand complex. Now you can see cysteine 797 is forming a covalent bond or covalent complex with your ligand molecule. Then other residues like methionine 797 is forming hydrogen bond interaction with the ligand molecule. Then E804 is also forming hydrogen bond interaction with your ligand molecule. If there are other pi interaction or pi cation interaction you want to visualize in your complex, then you just have to click on this button here, display pi cation or pi stack interaction, and it will show you all the pi or pi cation interaction between protein and protein as well as protein ligand molecule. So this way you can visualize our covalent docking results. If you want to compare the generated covalent docking results with the co-crystallized ligand molecule, then you just have to import the co-crystallized ligand molecule also in the workspace panel, or you can see there are multiple poses in the generated table, and they are quite superimposed. Like you can see over here, the generated pose is quite superimposed with the co-crystallized pose. And from visual analysis, you can say you are able to reproduce similar pose to your co-crystallized ligand molecule. But if you want to calculate the RMSD difference, you just have to go to tools and go to analysis, RMSD, chemical, and you can have the first molecule always will be automatically selected your co-crystallized ligand molecule. You have to select the second molecule which is present in your grid map, which is ICM. So you just have to select that particular M molecule and calculate the RMSD difference between generated and the co-crystallized covalent ligand molecule. So now you can see, and the co-crystallized ligand molecule is and the difference between these two molecules, generated pose and the co-crystallized ligand molecule, is 0.89. So we can say we are able to reproduce similar pose to our co-crystallized ligand molecule by using covalent docking in Molsoft. Similarly, if you want to generate the 2D interaction diagram for this particular complex, you just have to right click and use this option here, tools, ligand 2D diagram. You can remove this auto detect and select the particular object for which you want to generate 2D interaction diagram. Also change the labeling from 1-letter code to 3-letter code, and also create a table with the context and just press OK, and it will start generating your covalent complex with the 2D interaction diagram for your protein ligand complex. And you can see like here also cysteine 797 is forming covalent bond interaction with the ligand molecule. Then this particular residue is forming hydrogen bond interaction. This particular residue is forming hydrogen bond interaction. And then rest of the residues is forming hydrophobic interaction with the ligand molecule. And similar information you can get from the table, which is generated along with the 2D interaction diagram, like here lysine, then methionine and covalent interaction will be shown in the last, like 797 is forming covalent bond interaction with the ligand molecule. So I believe this is it from the today's session. If you have any questions, you can ask me.
Yeah, thank you so much, Dr. Khan, for the wonderful presentation and demonstration. Now we will move to the Q&A session. So if you have any questions, you can add them into the Q&A section. And meanwhile, I will address the questions we have already received. So the first question is why the target is always nucleophilic.
So far, whatever covalent inhibitors has been designed or approved, they are only targeting the nucleophilic residues. So these nucleophilic residues which are present in the binding site will be targeted by electrophilic warhead, which can form easily covalent bond interaction with the ligand molecule.
The next question is, can ICM handle multiple covalent reactions in a single run, or it
Yeah.
needs to be run separately for each reaction type?
Mm, no. If you have a single reactive residue, a single reactive residue, let's say cysteine, and you want to add multiple reaction transformation, then you can add. You just have to like go to. I will show you, just a minute. You just have to go to docking. So the docking, then covalent docking, one reaction is selected, then another cysteine reaction you can select, like the here we have selected acrylamide. Now I can select cyanoacrylamide also, acrylonide also, then other cysteine reaction also, and I will add all these reaction to our project. Now whatever ligand molecule I will be docking, if this particular warhead will be present in those ligand molecules, then it will form a covalent bond with the target protein. If this particular warhead is not present in the ligand molecule, then it will not form any type of covalent bond interaction with the ligand molecule. So you can use multiple reaction also at one go with the same residue. Okay.
The next question is, for new ligand, what is the command to determine the presence of reactive groups in it so that we know covalent docking is needed?
So a reactive warhead, you have to understand the reactive warhead as well as the reactive residue. Basically, you have to understand the protein of your target protein, whether it is having a reactive warhead in that particular reactive residue in the binding site. If that particular residue is present, like only the residue is present, then you have to determine the reactivity of that particular residue, whether that particular residue is able to form a covalent bond interaction. If that is also confirmed that this particular residue which is present in our binding site is able to form a covalent bond interaction with the covalent bond is having a reactivity, then you can try different warhead like acrylamide, then boronic acid, then there are several warheads which are, some are a reversible warhead, some are irreversible warhead. You can try them and see by which warhead your ligand molecule or your reactive warhead is forming a stable complex.
Okay. The next question is, can we view the interaction in 2D view in ICM?
Yes, 2D view. So we have generated the 2D interaction diagram as just of now, just I have shown. So this is a 2D interaction diagram only. So this is a 2D view. We can see how even you can double click and edit this particular diagram as well. You can zoom in, let's say zoom in, and you can move this slightly over here if you want to move these residues, any other residue. So you can move all these things, and you can just file, save and quit, and it will show you all this that particular modification also. So you can visualize a 2D interaction diagrams.
Okay. Now the next question would be, what are DFT calculations?
So DFT calculation is not available in Molsoft, which is density functional theory. If you need a high accuracy in formation of covalent bond, then you have to perform this quantum mechanics as well as molecular mechanics simulation. In this is required only when you don't have a data about the reactivity of your warhead and reactivity about your ligand molecule or warhead or reactive residue. In that case, you have to perform those type of calculation, DFT, which is not available in Molsoft.
Okay. The next question is there any scope to analyze charge transfer reaction energies through ICM?
Sorry, I didn't understand the question.
I'll repeat, Dr. Khan. Is there any scope to analyze charge transfer reaction energies through ICM?
No, I don't understand that. Sorry.
Okay. Actually, we have many more questions that we can address, but because we have a little time foundation, we will get back to all the unanswered questions through mail. And thank you all for the wonderful questions coming through the session. We appreciate your active participations. Unfortunately, we'll have to wrap up the session right now. But please be rest assured, all the unresponded questions will be reviewed and our team will get back to you via mail with detailed answers. With that being said, we have come to an end for today's webinar. On behalf of Advent Informatics, I, Alok Kumaja, sincerely thank Dr. Pathan Mohsin Khan for sharing his expertise and delivering such an insightful session. A big thank you to our audience for your time, engagement, and thoughtful questions. We look forward to hosting you in our upcoming events. Have a great day ahead and goodbye.